Hello, everyone, and welcome to the podcast, A Journey Through Nature and Science. I'm your host, Don Murphy. And boy, do we have a great episode for you today. We have with us Steve Gschmeissner, who has been, and I'm sure I just butchered his name again, who has been imaging microbes, cancer cells, and invertebrates for about 50 years, if I'm not mistaken. He's crafted an extraordinary array of more than 10,000 scanning electron microscope images. He joins us on this episode to discuss his craft and tell us how he captures these remarkable images. And we're going to share some of these images during our conversation. For those of you listening to the audio only, you can go to Steve's website at theworldcloseup.com. That's theworldcloseup.com. He has a beautiful gallery there of all these images, some of which we'll share with you here. And then we'll also have a blog page on Steve as well at natureandsciencepodcast.com. And you can go there, check out the blog page and more information about these absolutely stunning electron micrographs. So Steve, welcome to the program.
Thank you, Don.
Well, Steve, why don't you tell us a little bit about yourself? How did you get started with these scanning electron micrographs? And was there something in your background as a kid or growing up that inspired you to want to look at the world that we cannot see with the human eye?
Well, at my age, Don, when we were kids, we didn't have computers. We used to spend 12 hours a day out in the countryside, basically wandering around. We'd leave in the morning, come home in the evening. So I developed a deep interest in all things natural from a very, very early age. I would spend hours trawling streams, collecting slow worms, studying birds, watching birds. And this gave me a deep interest in the natural world. So when I actually went to school and did my O-levels and A-levels, it was natural for me to study zoology and botany. And from there, again, I went on to university to do a degree in zoology. So throughout my life, I had this deep-seated interest in everything to do with nature. When I left university with a degree in zoology, then the reality hit me that as a zoologist, you're not really qualified to do a great deal that's pure zoology. So I was very fortunate to find a job at the Royal College of Surgeons in England, working in the electron microscopy department. And that was a fantastic place to work. It has great museums and great historical significance, and I met an amazing number of strange and famous people there. And I actually worked there, I think, for 25 years, but that's when I was first introduced to the wonders of electron microscopy. A lot of electron microscopy units are service units. They're very expensive electron microscopes. I mean, modern ones can cost £250,000 to £500,000. So a lot of research labs can't afford them. So you'll have a service unit where the research labs can go to and have work carried out by this specialist unit.
So tell us what exactly, let's start off at the very basics. What is electron microscopy, and how does it differ from looking through a regular light microscope?
Essentially, light microscopes, I'm not a physicist, but I'll do my best to describe the difference. Light microscopes are limited in magnification by the wavelength of light. So the very best light microscopes will magnify up to maybe 1,000 times, and there you're looking at cell level, but no more than that. The wavelength of electrons is tiny, so you're not limited in magnification. There are limitations to how you use them because they will only move in a vacuum. So that's why if you see an electron microscope, it's this great huge column that you see, but they have the ability to magnify up to, well, theoretically, 1 million times. In real terms, you're not using those sort of magnifications. So essentially, an electron microscope takes over where a light microscope finishes.
Understand. Yeah, and it's the wavelength that's extremely important because that's how you see something is from the wavelength of light being reflected back from a
Yes.
particular image, and the smaller the wavelength, the smaller the image you can see.
Yeah, the higher the resolution you can
Higher
achieve.
exactly. And that's absolutely fascinating for those of you watching this video coming up on the screen in just a minute, you'll see an image of an electron microscope and exactly what it looks like. And then you'll also be able to go to our website and find out a little bit more information about how an electron microscope actually functions. So, without further ado, we're going to look at some of these images, and that way you can explain exactly the process that you use. And I'll bring up here on the screen for our viewers to see some of the images that we talk about that we're going to discuss here. So we'll start off with this head louse, and why don't you describe how you got this image? And I mean, this is just absolutely fascinating. I mean, this louse is so small that we would never ever see it with the human eye at this particular resolution. So why don't you tell us a little bit about this?
I mean, this is a relatively simple specimen to achieve because of the incidence of head lice or nits that school children tend to suffer from. So there's not a shortage of these sort of specimens. I think the important thing is that insects and a lot of invertebrates that have an exoskeleton have this fine detail so that they make perfect samples for scanning electron microscopy, which is what I specialize in. And here you can see the head louse is hanging onto a hair. And that's why it has
Mhm.
those big claws specifically. And you can't see in this section, but it's also flattened so it can move in and out the hairs very successfully on our heads.
Amazing. And so I know these, and we'll talk about colorization in a little bit more detail. Is this the actual color of the head louse, or how did you decide on this particular colorization?
I think the important thing is to start off by saying that color is a function of light. So electrons don't produce colored images, they produce black and white images. So if you were a scientist and using a black and white image, you would use it as a black and white images. You wouldn't alter it. But you know, we live in a colored world, so people want to see colored images. So the natural step for me is to color the black and white images, and you can color that in a variety of ways. You can color it naturalistically, which perhaps this is, and these little nits are that sort of color. You can color it artistically, and then you can do it any color you like. Or with some of the images, you will sort of color them functionally, where you will assign a different color to a different part of the image to help explain it. So there's a variety of ways of coloring, but there's no restriction to how you do it.
So you take this scanning electron micrograph, convert it to a digital image, is that correct? And then you use a photoshopping application or something of that nature to do the colorization?
Yeah, I mean, that's exactly correct. I mean, as you're aware, I'm sure, Don, when we started off in the laboratories, digital didn't exist and everything
Right.
was done on a plate or a roll film. The introduction of digital imaging to microscopy is a huge benefit because you could then take lots of pictures and select what you wanted from those pictures. But yes, it would go into, I do my colouring in Photoshop essentially. I'd
Right.
quite like to mention at the moment what's happening now is that with AI, I'm just starting to investigate using AI to colour images and getting some quite exciting results.
Mm-hmm. So when you use AI, what do? Do you tell the AI, you say, okay, AI, color this image and make it look good? I mean, what do what's the
It's
methodology? What do you do with the AI?
there isn't a methodology in the sense in that most AI, current AI, colouring AI, is designed to cope with what you would call a conventional image. So it will look at a person, recognize it, and color them whatever colors, natural trees, clouds. So with my images, it doesn't recognize them at all. They're too specialized. So it will have a go at coloring them in
Hmm.
a way that is arbitrary. And then you'll get a variety of options of colors that you can look at and see if any of them work.
Uh-huh. I see. Well, that's interesting. Hmm. I wonder if we'll get to a point where, AI and machine learning is so sophisticated that it'll be able to merge with a scanning electron microscope and I'll say see in quotes, the image in color. If you know what I mean. I'm just wondering if there will be a time when we'll get to that point technologically.
I think that the restrictions on that is that in science, pure science, people want the original black and white image. I know now that a number of I hope I'm correct with this. I think I am. A number of photo libraries will no longer accept AI generated images. I
Hmm.
like to think I'm doing something different because I'm starting with an original image and I'm just adding color to it, which is what I've always done. So I think we're in very early days of AI and no one really knows what will happen. I think we're going to see massive changes over the next few years with what AI does.
I think you're absolutely right. Well, let's go on to the next image. This is another fascinating image, and I'm sure a lot of people can relate to this, having gone through the pandemic. So this is a SARS-CoV-2, and the images of the virus are actually the buds that are in red here. You want to talk about this image a little bit? How did you capture this and decide on the colorization?
I, I think the thing about viruses that I love for what I do is that you can't see them down a conventional microscope. You can only see them down an electron microscope. So they're an exciting subject for a microscopist to look at. And I'd always wanted to look at viruses. And I'm very fortunate to work with a group from the University College London, run by Professor Greg Towers, who's been a great collaborator with me. And we've looked at a number. We had looked at a number of viruses before COVID arrived, and it was just a normal progression to actually start to look at COVID when it arrived here. And most viral work is done on cultured cells. So this is a cultured cell that's been infected with the virus, and that's a single cell budding out millions of viral particles. I mean, viruses
Mhm.
are fascinating anyway.
Oh, yes.
Conventional definition, they're not even living, but look at what, they've killed more people throughout history than all the wars put together. So, they're a great subject for me to look at, and I always enjoy looking at viruses.
Yeah, it's one of the reasons I got involved in science in the beginning. I mean, when I was asking you what started you off on your journey, for me, it was reading Watson and Crick's book on
Yeah.
the discovery of the structure of DNA, and that started me on my journey. And then viruses, Dr. Gallo and others who were early pioneers in researching um, viruses were a big influence on my life. You mentioned something really important our listeners should understand is that you cannot see viruses conventionally, and conventionally or even looking in a light microscope. And so their discovery was indirect. And I'm not going to go into a huge science lesson here, but the discovery of viruses and the fact that they even existed was discovered indirectly, and it wasn't until the advent, as you say, of the scanning electron microscope that we were actually able to see what a virus looked like and understand their structures. Um, this is amazing. It's important in a social sense, and I'd like to get your opinion and your comment on this. I mean, there's still a lot of people out there that don't think viruses exist and thinks, this is all a hoax and there's no such thing as a virus. You can't see them and it's all manufactured. But to what extent has electron microscopy and this visualization been able to help with the communication of science, facts, and information to people? And have you been involved in that at all?
I think what I like to think that I do, and this is since since I retired from what I call pure science and working in a lab and becoming a freelance microscopist, is to make this sort of scientific image available to the masses so everyone can access and and look at these images. I think in the past a lot of scientific imagery stayed within science, and the only other people who saw these images were science. And when I started this work and started working for the Science Photo Library, and they said to me there's a big demand for scientific imagery, I would have never guessed about the demand for this type of imagery and the interest that people have in seeing things like viruses and things that you can't visualize any other way. I mean,
Hmm.
as far as not believing about viruses, I've looked at, I don't know, 10 or 15 different viruses. They vary in structure. I mean, it's there before me. I have no doubt. It's
Hmm.
absolutely, if I think if you're a scientist and you have a scientific background, you don't doubt the existence of viruses, especially when
Right.
you've looked at so many that I have.
Right. And of course, if you get sick, that's some other evidence as well. It's interesting that you say this. I was talking to someone yesterday on another podcast, and we were talking about that there really needs to be a Neil deGrasse Tyson of biology and biochemistry to get these images and this information out there because people see stars, they've looked at them forever. We've had telescopes for centuries now, and people have been able to see galaxies and nebula. And of course, now with the color imaging and the digital imaging that we have, people are even more fascinated. And as you say, now that we have this kind of imagery that people can actually see what's going on a microscopic level and see inside the cell, I think it'll give people a deeper appreciation for life and for what's on inside. I mean, the microscopic world is just as vast as the universe out there, just as vast as all the galaxies in the universe, if not even more so. And so I think you're doing a great service getting these kind of images out there.
Thank you for those comments, Don. I'm st at 75. I'm still enjoying what I do because when I sit down at the microscope, I never know what I'm going to see. I can see something someone's never seen before or never shown before, and that's the challenge for me to we've recently looked at some new viruses that have never been visualized before,
Mhm.
so we're producing images that didn't exist before and making them public.
All right. That's fascinating. Well, let's go on to the next image here, which is another fascinating image. This is a bacteria from human fecal samples, and most people now understand the importance of bacteria in the gut bacteria, and a lot of medical diagnoses are done by looking at human fecal samples. And here's a really good example of what you can actually see. You want to talk about this for a minute?
Again, I think you're absolutely right, Don, that the biome, especially the human biome, I think over the last 5 to 10 years, people have realized how essential bacteria are to us. We have more bacteria in our bodies than cells, and they don't just control what's going on in your gut, but people now believe they are responsible for neurological things, even for emotions.
Hmm.
Every site in your body that you look at, and I've looked at most of them, has its own little bacterial biome, if you like. So not only are there millions or billions of bacteria in the body, but if you look at someone's umbilicus or belly button, you'll find a little colony of bacteria there that are not found anywhere else on the body. So again, they're incredibly important to us, and without them, we wouldn't exist. We couldn't live without our bacteria.
Right. You talk about relationships and connectivity in the world. It's one of the things that most of my listeners now know that I left the medical profession and became a park ranger, and it and worked in the environment. And it's amazing the connections that you end up understanding and seeing from the medical world to the broader world out there that I worked in the environment. Everything is connected, and we all life depends on other life. And images like this, I think, as I said earlier, create a deeper understanding when people can actually visualize and see these kinds of images and know that this is what's going on in the human body. I think it's extremely enlightening.
Yeah.
The colors on this are beautiful. How and why did you choose these colors?
I actually didn't use these colours.
Uh-huh.
The photo library I work for, which is like my commercial agents, if you like, they have an in-house colorist called
Hmm.
Caesar, who basically is based in Colombia, but he does beautiful colouring. He
Hmm.
What happens is you tend to get fixated in how you colour as an individual. So this is why I'm interested in the AI. But he colours in a beautiful way, and this is actually one of his coloured versions. I think essentially you're just colouring the different types. You have the two main types of bacteria there, the rods and the cocci, the round and long things. But it's the
Hmm.
subtlety of colouring, that makes it a nice image, and it's visually interesting.
Right. No, absolutely. Well, it certainly has its counterpart with the coloring we've seen from the Webb Space Telescope. Everybody's fascinated with those beautiful images that we see. And I think NASA's done a pretty good job in their information department communicating how they do the colorization of many of the images that have been sent back from outer space, if you will. So this is inner space colorization, and we have outer space colorization, and they're very similar. This is
I think we're
absolute.
going from one extreme to the other here, from
Exactly.
stars down to viruses.
Exactly. Oh, I think this is one of the coolest images. What is this?
This is a diatom. I love diatoms. I've taken hundreds and hundreds of pictures. They're basically microscopic algae that have a silica shell around them. And the beauty of them is that they have this incredible fine detail, and they're hugely pleomorphic. There's hundreds and hundreds, if not thousands, of different species out there who all look different, all have this microscopic detail. So, if you like, they're the perfect subject for scanning electron microscopy. And I never get tired of looking at them.
Yeah. This one's absolutely beautiful. Well, it reminds me of an Art Deco couch or chair. I've seen some Art Deco chairs that look exactly like this. So I bet there are some architects and furniture makers out there that can look at some of these images on a microscopic level and get quite inspired about what they are able to create.
Well, actually, one quite famous designer, clothes designer called Iris van Herpen. I think she's Dutch. She actually did a clothes collection based on some of my SEM images. I can't remember what it's called. I
Mm,
think it's actually called Micro, but she used some of my images to, I think she 3D print some of her
mhm,
dresses. But that was one of the more stranger use of my images. But no, diatoms are beautiful, and if you look at them, they often look like other things. I've got ones that look like perfect little handbags, and
mhm,
they're interesting on a number of levels. Apparently, they're more important than the Amazon rainforest for the environment, for the O2 and CO2 balances that go on there. You
mhm.
know, they're one of the things that some of the filtering whales eat in vast numbers. You get huge blooms of these
Mhm.
diatoms that you can see from space.
It's amazing. So, from a business point of view, are you able to commercialize any of your images? And like the lady that did the dress designs, or I mean, this takes a lot of skill and creativity on your part. Everybody can't do this at all. So, I'm wondering from a business point of view, can you capitalize on the creation of these images? I know these things occur quite naturally in nature and they're part of nature, but the creation of the images and being able to show what they actually look like, take your ability to accomplish.
I have a commercial agent, Science Photo Library, where all of my images exist. I
Hmm.
personally don't deal with that side of things, but there is a huge demand for the images. I can't give you numbers, but I think last time I looked, something like 50,000 of my images have been used worldwide. So
Hmm.
that gives you some idea of the demand for the this type of imagery, and it's not just for science. It's used as people have it as standalone artworks. It's been
Hmm.
on album covers. It's it's used, well, as you know, they were used on some USA stamps. So
Sure.
it can be used for multiple media, which I find exciting. You know, that it's not just used for science, but used for all sorts of other things as well.
Yeah, sure does communicate the beauty of the world we live in, and again, as I said, give you uh, a deeper appreciation for the connectivity of all life and the importance of maintaining that life and respecting that life. Here's the tardigrade, and there's a whole story behind the tardigrade from a science fiction point of view, which I'm not sure you're aware of. Maybe you are because of this image. I bet it was used maybe in some of the science fiction movies that have come out. But why don't you talk about this image a little bit?
The one that I remember, I think, is on one of the Star Trek episodes, the TV
Right,
series, that they have a huge alien that's actually a large tardigrade. I don't,
right,
I think it's a GI generated image that they use, but it was just amazing to see. I mean, they're incredibly fascinating, as I think you know, Don, and a lot of people do. I think they were sent into space. You can they stand up to radiation, to desiccation.
right.
I mean, they're called, I think, moss piglets or water bears because they're microscopic. But if you look at them, they have these little claws, and,
Right,
and they're cute. I think this is why the people like them. So,
right,
they're again, and at light level, you can see them and you can see them moving around, but you can't get this sort of resolution you can with
right.
the electron microscope.
Where did this image come from? Where do these tardigrades
I think
live?
I
Where
collected
did
these. I collected this one myself. I think they're called moss piglets. So one of the, I mean, they exist all over the place. There's marine ones, you get them in fresh water. The marine ones are fascinating but quite rare. But if anyone goes out and collects some moss and squeezes the water out of it and puts it under a light microscope, not only will they see huge numbers of protozoa and goodness knows what else, if they're lucky, they'll find water bears
Mm-hmm.
in the moss, hence their name of moss piglets. I don't know where the piglet comes from, but
I guess maybe the size and the shape. Well, I hope some of our viewers. I'll put a link on our website to the Star Trek show. These tardigrades not only lived in space, but there were these things similar to mushroom mycelium that they traveled in. So there were actually space mycelium that were universe and galaxy large, that were basically highways that these tardigrades, like creatures in the science fiction show, traveled in and lived on. And in this particular episode, the captain and engineers figured out how to adapt that mycelium highway and the way these tardigrades traveled to their warp engines so that they could move huge distances very quickly in space. It was a fascinating science fiction show that merged or juxtaposed the very, very tiny and microscopic with the with the vastness of the universe. I thought that was quite clever. This is really neat. Wow, what a beautiful image. And so here we go with a cultured cancer cell. What can
uh,
you tell us about this image?
after I stopped working at the Royal College of Surgeons, I went on to work for Cancer Research UK, which is, I think, the biggest charity in the UK. So I worked there for 15 years again, uh, in the late stages running the service unit there. So I came into contact with lots of different research projects associated with cancer. I've tended to be, I don't know what you would call it. I like to do everything I can possibly under the microscope. So over the years, I've worked through a whole range of different cancer cells, and these are cultured cancer cells, which simply means they're grown in the laboratory as a mechanism of studying cancer. And this one is undergoing something called programmed cell death, which is called another name for apoptosis, where all these blebs or vesicles are an early stage of cancer cell death. And it's interesting because I recently did a project looking at drugs that chemotherapy drugs that actually kill cancer cells, and we looked at this in the laboratory, and we followed the stages of normal cells all the way to complete cancer cell death. And this is, if you like the halfway stage where the cell's just starting to undergo changes that will eventually lead to death.
Hmm. Well, it's extremely important for scientists to actually be able to see what's going on physically. It's one thing to understand things biochemically, but when you can merge the biochemistry and the physiology with actually being able to see what's going on structurally, it's extremely instructive, and you discover things that you never knew were there. And this kind of, image is essential
I think you're absolutely correct there, Don, in that I think SEM on its own is quite limited, but when it's used, as you say, as part of a project, you're just looking at results in a different way, and you will often find things and see things that perhaps are unexpected.
hmm Yeah, absolutely. And it's a beautiful image as well. And here is kind of the final image, this midge larvae. And where was this? Did you collect this yourself as well?
I mean, this is an example of why I love scanning electron microscopy. I actually have a couple of allotments where I grow things, and I noticed on my raspberries I had a fungus growing, and I thought, well, that might make a interesting image. And when I actually came to look at it in the microscope, unbeknown to me, there was this little midge larva actually feeding on the
Hmm.
fungus, which is the green here. So it's again why I enjoy what I do. You're never quite sure what you're going to find. So I take a sample to look for something, and I found something unexpected, and again something that produced, I think, a lovely image.
Yeah, it's absolutely beautiful as well. So how do you access these electron microscopes? So when you collect this, where do you go, and how do you gain access to these expensive machines?
Uh, if you like, this is one of the limitations to my work. I was very fortunate to have a colleague who gave me access to a microscope for a number of years. Because they're so expensive and so large, they are limited in their access. But I actually pay for time. I hire the microscope, if you like, again at the University of London. So I buy time so I do all my preparation, have all my samples ready, and then go in and use specialist preparation equipment, and then the microscope. I think, as we mentioned earlier, the onset of digital is a massive help 'cause it allows me to take a lot of images in a relatively limited time.
Mhm. So when you collect a sample like this, do you have to do particular preparation or care between the time you collect it and the time you end up at the microscope itself to do the imaging?
Yeah, it's very critical how stuff is prepared. If you think about an electron microscope, as I said before, it has this huge column, but it's actually under a vacuum, and you're bombarding your specimen with electrons. So neither of those are ideal for anything biological. So the steps are relatively straightforward in that the sample would be fixed, which means it's preserved, it makes it stable. It's then
Hmm.
dehydrated, removing all the water. It's then dried in a very specialist piece of equipment so it doesn't distort. And then it's covered in a very fine layer of gold, which is there to reflect the electrons and actually produce the images. If you didn't have the gold, the specimen would charge and heat up. So it's a critical and specialized preparation, but it's pretty standard.
That's just amazing. But I mean, it does take skill and know-how and ability, and I mean, it's a craft in itself. Not everybody can do this. It's just absolutely beautiful and remarkable what you're able to create here. So what's next for you? What are you going to do next? Are there specimens or images you have never seen or taken before that you're on the hunt for?
To be honest, I'm slowing down because of my age,
Uh,
but I'm always working on different projects. At the moment, I'm working with a group in the University of Florida on diabetes, and we're looking at not
hmm.
scanning electron microscopy, but transmission electron microscopy, and we're producing some beautiful images of islets. It's a group who are working on diabetes.
And
And
islets are the cells that produce insulin, is that
yeah,
right?
basically insulin producing cells. So we produce some sort of unique and I think very beautiful images there. I'm working with a group on chronic rhinitis at the University of Vienna
Hmm.
and continuing the viral work. We're hoping to look at some other viruses with with the group in again at University of London. But I never know what I'm going to do. Sometimes I'll be approached to carry out a project, or something will occur to me that I'd quite like to do.
Right.
So it's an unknown future for me, really, to be honest, Don.
I understand. Well, you mentioned the transmission microscope. What's the difference between that and a scanning electron microscope?
I mean, can I share an image with you? Are we able
Sh-
to do that?
Sure, if you can, let's see. Let me stop sharing this image and maybe we can bring it up on the screen.
I'll do the one of the eyelet, which will demonstrate, I think, really nicely.
Okay, I'll stop sharing this one and maybe if I can see your screen, maybe we'll be able to do that. Let's see.
So do I just press share now, I wonder?
Yeah, you should just be able to press share,
Share. Sharing is not
and then if you have the image on your,
in request. Request has been
I
sent.
got it, so I can allow.
Yeah. And now
And it's the keynote electron. This one. Let's see if
uh,
this is the one.
so you okay.
Oh, that's not the one. Hold on.
Can you see my screen? I wonder, or do I need to share it with you? Am I making things complicated?
No, no, not at all. Hold on just a minute. We can because I just gave you permission. So why don't you do that again? So you have an image. So if you'll press
Share.
share.
Okay, here we go. So screens, yep. Share. Okay, so I should be able to screen sharing. So I wonder if I can open desktop. I don't know if this will work. No, I've done something stupid now.
Okay, so I'll tell you how to do it, okay? So if you will go in first and open up the image and just put it on so that it's on your screen or on your desktop. That's what you want to do.
Okay, there it is.
There it is.
Can you see that?
Yes, and our viewers should be able to see it too. There we are.
Okay, so this is a whole normal eyelet, and I'm just. Can I just pause 1 second? I just want to make sure that I give credit to who it's from, and I don't want to get his name wrong, so I need to. It's oh, I can't remember. I'm sorry. Okay, let's ignore that. I just wanted to give credit to the prof who's
Well,
uh,
that's okay because you can send that information to me
okay,
and
that's fine. So
I
you
share
can see
that.
this. You can see this is totally different type of image. This is two-dimensional, and electron microscopy is divided into two fields: scanning electron microscopy, which I specialize in, have been all the images that we've looked at, but also transmission electron microscopy, and this is where you can actually section a material. You cut inside and you actually look inside the cells and you get information about the cells and what's going on within them. And I don't know if I can zoom this, but the beauty of it is you can actually zoom a cell and look at mitochondria, nucleus, and these are granules. I think these are the insulin granules, but I could be wrong. So essentially, we're looking at alpha and beta cells here.
Mhm.
I don't know if how to zoom out. I need to go fit on screen.
Well, the reason I was asking you about the difference, that's we used transmission electron microscopes years ago when we were looking at when we were studying myasthenia gravis and trying to understand whether or not the synapses were being destroyed or not. And we could take uh, pictures like this,
Yes,
transmission
essentially.
electron micrographs, and we could see actually the destruction that was being caused by the body's own immune system. And
So we will have equivalents of this, Don, from diabetic patients, where you'll see the changes. A lot of these beta cells will become abnormal and be
mhm,
lost.
right.
So I always think transmission and scanning work well together. They're good bedfellows. One shows one thing and one shows another. But to me, again, I'm biased, but this is a beautiful image because an image like this doesn't exist anywhere else. I think we're the first to have done it, and I only take credit for the colouring, the actual production, and the TEM were done by a group in Denmark. So they've contacted me to take this to this level, if you like, to this colouring level. But again, it's just a way of visualizing something that you wouldn't normally see.
Yeah, what a tremendously important tool, both beautiful and practical in terms of especially marrying the structure and function so that you can really see from a lot of times the structure and understand the function of what's going on. It's just absolutely fascinating. I tell you. Well, we're just about out of time here. And Steve, I can't tell you how much I appreciate you sharing these images with you. And I'm excited about getting some of these images out here in the United States so people can see them again. They can go to your website as well to see some of these images. And I'll certainly be posting it on our website as well. So Steve, thank you for joining me on this episode of Nature and Science podcast.com. And I really hope we can stay in touch and communicate with each other about these things, because I think I'm going to have lots and lots of questions that come up as a result of this episode.
Thank you, Don. I've enjoyed it. I don't often get to discuss my work with people, so it's been a pleasure for me as well. Thank you.
All right, thank you. Well, listen, you have a great rest of the day, and I'll let you know when this episode is live, and I'll make sure that I share it as broadly as I can, and you do the same on your end.
I will do. I look forward to it.
Okay. Thank you, Steve. Take care.
Thank you.
Bye-bye.
Thank you. Bye-bye.
I want to thank you for joining us on this episode of A Journey Through Nature and Science. On our next episode, we're going to be joined by Dr. Kenneth Monahan. Dr. Monahan helps stroke patients recover in their own home and tells caregivers exactly what they need to do to relieve some of the stress of helping caregivers. So please don't forget to hit the like button and leave a comment if you enjoyed this episode. And you can always go to our website at natureandsciencepodcast.com and see more images of these beautiful electron micrographs. So as always, stay safe, share happiness, and remember, never stop questioning. Curiosity has its own reason for existing.